आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सोल्यूशन पाओ। डाउनलोड नाउ।
Hello everyone, so the question over here says that explain the importance of first is ORI, this is not IORI, it is ORI. B is ampicillin resistance, C is ROP in case of the E. coli vector. So first of all you should know the full form of these three sentences. ORI is origin of replication, origin of replication. This is the point that is present in case of the plasmid of the E. coli bacteria from where this is the point basically from where the replication start, from where the replication of this plasmid start. So if we are going to incorporate our gene of interest or our DNA of interest over this point, then this DNA will start the replication when if it is present in the host cell. All right, and this ORI is also responsible for controlling the copy number, also responsible for controlling the copy number of the replication. So if we want high copy number of that DNA of our interest or gene of our interest, then we should incorporate this DNA into a plasmid which has got high copy number. All right. Second is ampicillin resistant. This is basically ampicillin resistant. AMPR stands for ampicillin resistant. What is this ampicillin? Basically, this ampicillin is an antibiotic. This is an antibiotic, and if this gene is present over here in this plasmid, it means this gene prevents the E. coli, that is, it makes the E. coli resistant against this ampicillin. So this E. coli bacteria won't die in presence of the ampicillin antibiotics. It means if this kind of the resistant E. coli is present, so this ampicillin will have no effect on that bacteria. And what is the importance from with if we incorporate our DNA of interest at this area, ampicillin resistance area, then we can differentiate versus recombinant, that is recombinant versus non-recombinant E. coli bacteria. Recombinant versus non-recombinant. How this will happen? If we are going to incorporate the DNA over this area, then what will happen? This particular E. coli bacteria will lose the resistance for this ampicillin antibiotic, and hence if we are going to grow them into a culture medium, this is the culture medium which consists of the antibiotic ampicillin. So the non-recombinant will grow in this culture medium. Why? Because it is already resistant, whereas the recombinant, which has become non-resistant to this ampicillin antibiotic, it won't grow in the culture medium, and hence it becomes the selectable marker. It becomes the selectable marker for the recombinant as well as non-recombinant plasmid bacteria. All right. Third one is ROP. ROP stands for replication of the plasmid. ROP stands for the replication of the plasmid. Replication of the plasmid. So basically this is the area where the replication of the plasmid take place. All right. So if we are going to incorporate our DNA of interest at this area, so what will happen? The replication, even the DNA will start replicated, or gene of our interest will start replicated if we incorporate the DNA in this particular area. All right. So these are the importance of the ROP, ampicillin resistance and origin of replication. Thank you.
क्लास 6 से 12 से लेके नीट, IIT, JEE मेंस और एडवांस के लेवल तक 10 मिलियन से ज्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।